Hi, I'm Maddie, and today I'm going to explain to you how the mutation of a single nucleotide within phenylalanine hydroxylase can lead to an individual being diagnosed with phenylketonuria. This sort of mutation actually happens quite a lot, and it can also happen in different locations along the phenylalanine hydroxylase protein. So this video is just a bit of a general overview of what can happen in actually a lot of different spots along the protein. So first we are going to discuss the regular production of a normal phenylalanine hydroxylase protein before going into the process behind the production of mutated phenylalanine hydroxylase proteins. So this is a basic DNA sequence. Everything in our body is coded for by our own distinctive DNA sequences. DNA sequences are almost identical in every human body, with each small change responsible for things such as like physical variations, including eye color and hair color. But these differences in DNA sequences can also lead to disease, and this is indeed the case for phenylketonuria. So this DNA structure, however, is completely normal. There's nothing wrong with this. These letters in the squares signify the four molecules which comprise the DNA sequence. So thymine, cytosine, adenine, and guanine. And the different combination of these four letters, or we call them bases, form the entirety of the codes necessary to create a fully functioning human. So from this point, through a biochemical process called transcription, these DNA molecules are read like a book by our body and copied into an alternative form called mRNA. So mRNA is a long strand of molecules which code for the production of proteins. This mRNA sequence includes coding regions known as exons, and these are visible in the orange. But mRNA also includes non-coding regions known as introns, and these are visible in the grey. But these orange regions are what supply the instructions to create proteins. These grey sections do not. So it's crucial that these grey non-coding regions are removed before any further processes can occur. So to remove these grey coding regions, what is known as a spliceosome practically eats up all of the grey areas and glues back together all of the orange areas. These orange areas are what form the spliced mRNA sequence. So this sequence is made up of only molecules that give instruction for the production of proteins. These molecules are then read once again by our bodies in order to derive the appropriate amino acid sequence. This occurs through a process called translation. This mRNA sequence gives instructions to form these amino acids within our cells. These amino acids then begin to link and fold and basically talk to each other to form what becomes the final protein structure. And it's only at this point that this protein is able to function effectively and work to break down phenylalanine into tyrosine. So this protein down here is described as being in its native state, meaning it's properly folded and assembled, and that way it can operate and be a fully functional protein. So now that we understand this process, these steps, let's move over and take a look at what happens when the original DNA sequence is mutated. So here we have the DNA sequence of a mutated individual, and you can see that where a G formerly resided, a C is now present. This isn't what we would necessarily consider normal, so this has caused an error within what is basically the primary line of the structure. But nonetheless, this sequence is converted into an mRNA sequence, ready to cut out all of the non-coding regions and sequentially then have the coding regions be pasted back together to form that long strand like we had before. But this pink region here is showing where the mutation in the DNA sequence has actually occurred. But as this code has altered the basic sequence of the DNA, this region is now unrecognizable to the machine that does the cutting and the pasting. So now when the spliceosome, that big red thing from before, comes along to cut and paste our mRNA sequence, the entire orange region here is going to be excluded. because the locations which should have been cut and glued are now unrecognizable in this region. So we now have a truncated, spliced mRNA sequence. And of course, this consequently leads to a truncated amino acid sequence. And this then leads to a truncated and thus not functional protein, as the native state of this protein, phenylalanine hydroxylase, is now no longer possible. So that is how people get phenylketonuria, as this enzyme is no longer able to function properly due to a disruption in its structure.